to speed up the development of new cultivars. It's a platform based on controlled temperature and constant light, which means 24 hours of photo period. And it allows up to 6 generations of spring wheat per year. It's suitable for wheat and barley and all year round. The aim of my talk today is to show how we can use the speed breeding system in breeding programs. So first, I will describe all the phenotyping methods that we developed in our team that are suitable for the speed breeding system. And then I will show how we can rapidly select for multiple traits at the same time using the speed breeding system and this phenotyping method. But before I jump in my talk, I just wanted to clarify the different phenotyping methods that breeders can employ. There is the phenotyping and phenotypic screening. Phenotyping is all about accuracy. It requires robust design. The methods are accurate, but it means that it's often quite expensive and low throughput. So it's good to characterize inbred lines, but it's difficult to apply in large population. Phenotypic screening is all about throughput, rapidity. So the methods are generally less accurate, but they're easy, cheap, rapid, so we can apply them on large population, especially in segregating population. Then, and this is what we're trying to do with the speed breeding system, precision and high throughput phenotyping methods. So we are trying to get as much accuracy as we can, but we still want the throughput, cheap, easy, rapid. So we can use the methods to both characterize inbred lines, but also for selection in breeding programs. The strategy that we employ is we first need to identify some good proxy traits. A good proxy trait is a trait that is correlated with a trait relevant for breeders, that have a high heritability, that you can measure at early stages, and importantly, that they correlate with the yield or yield components in the field. And then we develop these methods, as I said, cheap, rapid, and as much accurate as we can, so they're repeatable across experiments. This is an overview of my talk. So I am going to present all the phenotyping methods that we develop for grain dormancy, root traits, rust, yellow spot, and crown rot. So this work was conducted by some students in my team, so I'm going to introduce their work. If you have any questions, please feel free to interrupt me, and Lee and I will take the questions. First, grain dormancy. Why grain dormancy is important, and what is it? Pre-harvest sprouting is when the seeds, as you can see on the picture, when the seed starts to germinate, when it's still inside the head. It happens in many parts of the world, but especially in northern New South Wales and in Queensland, because the harvest period coincides with the summer rainfall. It can have very bad effects on yield and quality. Grain dormancy is when the seeds do not germinate, even if the seeds are placed in the optimum condition for germination. So that's a trait relevant for breeders. And typically, dormancy is screened by germination testing of harvest ripe grain. But in the field, it's quite complicated. Grain dormancy is controlled by many QTL, so it's highly influenced by environment condition, and it's often limited to characterization performed late in the breeding cycle. So the idea was to do it in controlled environment conditions using the speed breeding system. So we would have a more uniform growing environment because we can control the temperature, the humidity, the photoperiod, but also because we could extend the photoperiod and accelerate the breeding cycles. So here are the main steps of the protocol that we developed. So the individual heads are harvested at the point of physiological maturity. We are using the collapse of the primary node. If you look at the picture, it's actually linked to the maturity, and it's just an easy way to detect when the plant is mature. And it just makes our life easier because it requires less skills. We then put the seeds in the oven for 48 hours. We thresh the spikes. We store at -20 to preserve grain dormancy. We then test for dormancy using Petri dishes. And then we count every day during 2 weeks the number of grains that has germinated. And we calculate a weighted germination index to be able to compare the measures of grain dormancy across environment. And the cool thing is that we are then able to dry and to store the selected seeds. And we found that between 70 and 85% of the seeds were actually able to re-germinate, which means that we can select seeds and continue later in breeding programs. Using this protocol, we made a first test. Can we use this protocol to characterize fixed lines? To do this, we used a couple of lines, some double haploid lines that were dormant, and some Australian cultivars that were released recently, but they were known to be non-dormant. Yes, please.
Are these red or white kernel weeds?
White. Red, white.
They're white?
White.
And we characterized this line in the speed breeding system. So we did 3 experiments, and in the field across 2 years. And this is what we obtained. So here is the grain dormancy. You have the double haploid lines here, and the Australian cultivars here. On the left is the speed breeding system conditions, and on the right, the field condition. And as you can see, the level of dormancy reached by the dormant and non-dormant were quite similar in the field and in the speed breeding condition. And we were able to discriminate the dormant and the non-dormant lines in the speed breeding system, which means that we can use this protocol in controlled conditions to characterize for grain dormancy. Test number 2, we tested if we were able to apply this protocol for selection in a large population. So what we did is we used a population from a cross of a dormant and a non-dormant line. So this is the distribution you would expect when you cross a dormant parent with a non-dormant parent. You would have a normal distribution. And then we just selected for the 100 most dormant. And we did 2 cycles of selection at the F2 and F3 stages. And so this is the result for one of the population. You have the germination index and the number of F3 family. And as you can see, we have this nice shift towards the dormant. So here is the germination index. So basically the lines here are dormant. So we were able to select for grain dormancy in a segregating population. And we had these results in the 7 crosses. So that's it for the grain dormancy. We showed that the protocol that we developed was could be used to characterize fixed lines and to select in segregating population. Also, the important thing is the previous work, the development of the lines to the F4, F5 stage was performed using the speed breeding system in only 18 months.
Might just add that what we normally see if we don't do selection is the opposite. So it's like very rare to find highly dormant individuals. There's a large number of genes.
So, just a question. Even though you're doing the high-speed generations, you're able to select, and in that case, you are harvesting and planting, so you're eventually and directly selecting for less dormant lines. You are getting a very high proportion of dormant lines when you're doing the speed breeding?
In this case, it's because we applied a selection. We selected for the most dormant lines at each generation, so that's why we had this shift. But if you do not apply a selection on grain dormancy, it shouldn't change.
It means that the cycle time is longer because we have to grow them all the way through to maturity. So it's about 10 weeks, sometimes 12 for late maturing times.
Okay.
Could you please in detail tell what is the temperature you are applying and the lighting conditions?
Lee, do you want to?
It's the same rule of thumb. We normally use 1722 night day with constant light. That's just a recipe that works for us. I'm sure you can manipulate things a bit more, and some people often use have a little dark period there, so it's an extended photo period. So it's just a matter of getting the recipe right to get good healthy plants is the most important thing.
Lighting, are you inducing far red is more for early flowering? What kind of lighting you are providing for early flowering?
We just use sodium vapor lamps in most of our glasshouses. You can use regular fluorescent tubes. That works. And like I said in my talk, day one, we also use spotlights as well.
24 hours?
Yep. Did you miss the talk on day one, or?
Yes, sir.
No?
My poster is not there. I missed it.
Well, they'll be all available so you can watch it.
So have you played with wavelength of light at all as far as triggering?
No.
All right, root traits. So that's the core of my PhD, so I would be more able to take the questions for this topic. So we talk about roots a lot during this conference, so I'm just going to go quickly through the introduction. But basically, deeper root system and a greater branching at depth should allow access to water, especially in deep soil, under drought, moisture, sorry about that, late stress. When there is no water anymore in the shallow soil, but there is still water at depth. Modeling has shown that any extra millimeters of water extracted during the grain filling could lead to an increase of 55 kilograms per hectare. And studies shown that the deep root system and great branching at depth was actually linked to the seminal root angle and number. So these two traits are good proxy traits to select for root system architecture. This picture is just an example of a study in rice. So they identify a gene linked, responsible for the root angle, and they cloned the gene and they integrate it into shallow cultivar. So if you look at the picture, on the top is the shallow cultivar, and on the bottom is the same cultivar but transformed with the gene. And they showed that with this gene linked to the seminal root angle, they were actually able to modify the architecture of the roots toward a deeper root system. And they also showed the correlation with the field, because this transformed rice had a better yield across 3 different environments. Phenotyping roots is quite a pain in the neck. It can be performed in the field or in the laboratory. So in the lab, generally the containers that are used are a bit too small to mimic the available volume in the field. And it's difficult to reproduce the field-like conditions. But at the same time, it's more precise because the conditions are more homogeneous. So it's more reproducible. It's avoid to have all the environmental interactions, and it's easier and more rapid. So this is the phenotyping method that we developed. It's very simple. It's based on clear pots. The seeds are sown embryo towards the wall and facing downwards. So when the roots are emerging, you can see them across the wall. The plants are grown for 5 to 10 days at 17 degrees. And then we can have access to the number of roots and the angle based on images. So this is the system that we developed. It's a revolving stand, and we take a picture of each individual plant, and then we stitch all the picture together. And then, so this is an example of one individual plant. So the first challenge is to identify the different roots. So you have the radical and the first pair of seminal roots. And then using a software, ImageJ, we are able to calculate the angle. So this method is high throughput. It's only 5 to 10 days. You can fit 600 plants per square meter. It's quite cheap. It's less than $2 for a pot, and it's really easy to set up. We are currently working on the software and an app to automatically detect the roots, to also automate the stand, so it will rotate by itself. And we also created a system of plates to organize the seeds in the plates to follow the randomization, so it's just easier to sew. Yeah, this is a video of one of our students, and she's just showing how she's doing it. So basically, she's preparing the seeds in the plates according to the randomization. So then when she's sewing, it's like she's doing. She just takes the seeds one by one, and she put in the pots. That's the speed breeding system. Taking the pictures and measuring the angle. So altogether, you can have access to the root traits in within 2 weeks. So that's quite.
Can I ask, like the roots at the seedling stage, do they really represent the root architecture at the adult stage? So is it the same?
Yeah. Well, so previous studies shown that there were a correlation between the angle and what actually happened in the field. I introduced the example of rice. They showed that it was working. For my PhD, I'm going to investigate this link with the field, but we have the trial this year, so I will know a bit more later.
Some of the, because we also, we're not sort of looking at the roots in isolation, and part of Cecile's work is to marry up this information on seminal root traits with yield data and also staging data and phenology. But interestingly, when we measure these traits, we're tapping into existing data sets, and we find very good genetic correlations with yield in water limited environments. So we know that the trait is very important. Making the connection Is it actually getting those roots deeper down in wheat? Still needs further work and validation, but certainly it's linked to yield, and that's pretty important. So.
There's good evidence in the literature that those deeper seminal roots are linked to a narrower root system in the field. That's where the barley, wheat, winter wheat, and we've shown the spring wheat as well in the past courses.
And also, are you focusing on the logic or the root anchorage strength or that sort of thing?
These are only seminal roots, and for logic it's usually the adventitious or nodal roots that are more important for lodging later in the season. So we haven't investigated the nodal roots because in our system the seminal roots grow down through the profile and extract most of the water, leaving the upper profile dry, so that when the nodal roots grow into it, it's pretty dry. So in terms of water extraction, they're not very important. There are others in Australia who are certainly investigating nodal roots in terms of their local lodging, especially for irrigated cropping systems in Australia.
But there also takes a lot longer to measure them too, in terms of time. This is a pretty fast and dirty way of measuring something that's pretty important.
About the soil, the angle you measure from the greenhouse condition and special soil you are using. But about field condition, the angle is same, or for my opinion, the field situation, the ankle and the root cannot grow like that. You selected by from the greenhouse condition, how you fit from field condition. I don't understand.
You mean the type of soil that we are using, if it's the same as in the field?
Because the soil situation is different in different part of world. For example, in my country, our soil is not easy to work like that. So if I try to fix that, your example to my work in my country, it's not easy to work like that. And the greenhouse condition, the water condition is okay, but to the field condition.
But again, it's a proxy trait. So we know that it's going to be different in the field, but what we're trying to have is just a correlation between what we observed in our controlled environment condition with a different type of soil and what actually is going on in the field later. The soil that we are using is a pine bark mix, and we use this one because it's very dark, so it's easier to detect the roots. But I mean, the structure and the volume of the soil is definitely different from the field. But again, we are just looking for the correlation with the field.
And what is the coefficient of variation among replicates that you use?
So, for example, you mean for the same genotypes? I think it was quite high between. We had a correlation between 0.7 and 0.8 between each experiment. So it was quite...
Do you have one single replicate every time, or do you have different replicates?
No, we do have 8. We usually use 8 reps. But 1 rep is 1 single plant.
Yeah, but between single plants, the root angle that you measure?
There is a lot of variation for one genotype. There is a lot of variation within each genotype, but then we are still able to differentiate the different genotypes. When we...
A high coefficient of variation?
Well, it means that because we are measuring each individual plant, we do have variation for the same genotype.
Okay. So this is an experimental error, or?
Because you know the seeds are different. They have different size. You have the measurement error and all this kind of stuff. So it's normal. You're not expecting to have the same. So we do have variation within the same genotype. But what is important is that we have more variation between different genotypes than within the same genotype. So we are still able to differentiate the...
It's a very sporadic trait. Like these roots just come out and seed, like you know. But but funny enough, that's still under genetic control of which angle they come out. But if you think about it, we're using 8 reps to characterize fixed lines, but in a field you've got like millions of seeds, and so we're looking at average effects in terms of angle. So if you shift the angle 10 degrees, it can mean big things underground.
All right, so with the protocol, we tried to test if we were able to characterize fixed lines. To do it, we developed a name population. It's actually 3 different name populations. We crossed the same founders to 3 different common parents, and we characterized for root angle and root number. So on the left is the root angle, and on the right, the root number. And each box plot corresponds to a name family. So we have 3 name families. And I guess what I want to show here is we had variation, high degree of variation. I think it was from 54 to 95 degrees for root angle, and from 5 to 9 for root number. And we had this variation within each name family, but also across the 3 different name populations. Sorry. The heritability was quite high, with O.64 for angle and O.70 for root number. So yeah, I had the question with the link with the field. So as I said, we don't know yet, I mean, as part of my PhD, but one of our PhD students, Hannah Robinson, she's working with barley, and she identified major QTL for root angle on chromosome 5H. And what she did is she had this population, and some of the lines were carrying the peak marker for this QTL, and some didn't. And she compared the yield in across 20 environments. And what she showed is that the lines that were carrying the peak marker for root angle QTL showed a significant yield advantage from 5 to 11, as you can see on the table, in 16 environments and across 3 years. It doesn't prove that there is a link between angle and final yield, but it's very exciting results, and it's very promising. Tester number 2, can we apply this protocol in segregating population? So I used 2 different crosses, a parent with a narrow root angle and a parent with a wide root angle. And I crossed them, and then, so this is the distribution that we would expect, and then I just selected for the tails by selecting 20% of the narrowest and of the widest seminal root angle, and I did it 3 times at the BC1F2, BC1F3, and BC1F4 stages. And this is what I obtained. So this is the distribution of the root angle. On the left is for the BC1F3, on the right for the BC1F4. The colors correspond to the 2 different tails. In pink is the narrow tail, and in blue, the wide tail. So I had about 500 lines at the BC1F2 stage, and then I just select for the 20% widest, narrowest. And then for each population, I kept a size of 270, but because of the cycle of selection, I ended up with 50 lines for each population.
Why do you call it BC1?
Because I did a step of backcross. Yeah. The idea was my narrow parent was not adapted to the Australian environment, and the wide one was maize, scout, or suntop, an adapted parent. And that's why we added an extra step of backcross to be able to study the distribution in a background that is a bit more adapted to the Australian region. So as you can see on the left, at the BC1F3 stage, the two curves overlap, and we're not really able to see anything. But at the BC1F4, we actually do have this shift between the distribution of the two tails, which means that the narrow tail had a significant narrower root angle compared to the widest one. So the clear-pot method that we developed can be used to characterize fixed line and to select for either wide or narrow angle in segregating population. Rust. So there are three types of rust: stripe rust, leaf rust, stem rust. The pathogens are rapidly evolving, and we are looking for adult plant resistance because it's supposed to be the more durable. Typically, rust can be assessed in planted rows in the field with the use of disease nurseries where plants are naturally infected by the pathogen, or using artificial inoculation. And the plants are assessed for resistance, usually late after flowering. If we could phenotype rust in control environment conditions, it could be conducted all year round. We could incorporate rapid generation advance, and it will also minimize the interaction with the field, with the environment. So briefly, that's the protocol that we developed. I guess what I want to highlight here is the fact, well, the idea was, why would we screen for one type of rust when we can screen for the three at the same time? So that's the idea, and that's the protocol we developed. So basically, the plants are growing in the speed breeding system for 3 weeks using constant light. So they're growing very fast. Then we inoculate stripe rust on leaf 3 or 4. Then the trays are put back in the optimum conditions to allow the development of the disease. For 5 days, the plants are assessed for stripe rust, and then they are inoculate with leaf rust and stem rust. So basically, we spray the leaf, and then we spray the stem. And then the trays are placed back in optimum conditions for the development of the disease. So that's the scale that we developed to assess stem rust and leaf rust and stripe rust. Lee, do you want to mention anything about this?
Yeah, so the idea is to evaluate these pathogens on different parts of the plant. So we're actually evaluating the stripe rust on the leaf 3 and 4, where you can detect those chlorosis and necrosis associated with adult plant resistance. And then we evaluate stem rust on the stems, where it should be evaluated, and leaf rust on the flagging flag -1. So, but even when we do get localized infection of all three pathogens, as you can see, I mean, sometimes you can tell the difference and you can differentiate susceptibility and resistance to each of the pathogens. But you know, we tried to avoid that. But I guess that picture was just to highlight that you do see these combinations even when they occur on the same leaf. But we try to score them on those different parts of the plants.
What's to stop the leaf rust going on the same as the stripe?
So the stripe rust, it goes on first. The stripe rust goes on first after 3 weeks of growth, and then we put them back in the speed breeding system, but diurnal light and cool night temperature to favour stripe rust development. But then 4 or 5 days later, we then inoculate with leaf and stem. So the stripe rust is already in the plant, and
You've already scored it at that point?
No. And it's just we need that cool temperature to set the reaction. It's very important the first few days after infection. And then the leaf and stem goes on top, and the plant keeps growing in those 4 or 5 days. So they produce another new leaf, and yeah, it's a bit of 5 days. It's good. It sort of allows you then to put the stem rust on first by sort of holding the rows of plants back with your arm and layering it onto the stems, and then at the end, then we go with the leaf rust over the top, and sort of just that sort of helps you focus the leaf rust on those upper leaves where you've got the stripe rust on the lower ones.
But you will get a little bit of that.
Yeah, you sort of just exactly. And, usually when a plant has rust or a high degree of rust on it, you don't want it, so you know.
Could you have perhaps reactions more exacerbated, like you're weakening the plant first with stripe rust and rust? It's kind of a
it's very intense. But from a plant breeding perspective, I guess I'm not a pathologist. Or something to do with pathology, but from a plant breeding perspective, you just want the healthy ones that don't have rust, and that's what we use it for. Yeah. It's also interesting to just, I guess, also keep in mind that these rusts occur together in the field as well.
Yellow spot. So we also developed a protocol for yellow spot. I guess we were inspired by the rust, that we could screen for 3 types of rust, and we just think about yellow spot. There are 2 different resistance: seedling, adult plant resistance. And I guess the idea was, are we able to screen for both seedling and adult plant resistance at the same time? So that's the protocol that we developed. Plants are growing for 2 weeks in normal conditions. Then we inoculate the plants with yellow spot. The trays are placed back in optimum conditions. We assess for seedling resistance. Then the trays are transferred for 2 weeks to accelerate growth conditions. We inoculate again, and we place back the trays in optimum condition, and we assess for adult plant resistance. So to validate the protocol, we had to test 2 things. First, do we have the same results when we grow the plants in normal conditions compared to accelerate growth conditions? And secondly, do we have the same results when we are using independent experiment and integrated experiment? Which means, in one case, we assess seedling resistance in an experiment, and then we are doing another experiment to assess adult plant resistance. Integrated is when you assess the same plants for both seedling and adult plant resistance. And this is the results that we get. So if you look on the right, so this is the response for this disease for a panel. We do have, so the top arrow is for the seedling response, and the 3 different arrows here. This one is for when it's grown in accelerate growth conditions, this one in normal conditions, and this one the integrated approach, so when the plants are screened for both seedling and adult plant resistance. So as you can see, the arrows are quite close to each other, which means that we get quite the same results. And the correlation, if you look at the table, so this is the integrated approach, was very well correlated with the when it was assessed independently, either in accelerate growth condition or in regular conditions.
Cecile, can I just add something? Sure. Just one of the tricks with dealing with yellow spot that we use is we don't culture the conidia. So we actually do a field increase of spores, and we collect them using a spore collector, and we store them at negative 80 same as you do rust. So we actually just set up your little tubes in the freezer, and you go and get a tube or two every time you do inoculation. You don't have to all this effort to culture plates and plates and plates to screen a large population. We've got actually like 10s, 20s, 30s grams of this stuff, and we just do an increase every year.
Has any of the yellow rust selections you've been validated? Yes.
Oh yeah, actually, I think it's Adnan's work, the validation of the rust or yellow spot in the field. Lee, do you want? Yes, Lee, do you want to? The validation of the leaf and rust and yellow spot in the field. Yep. I don't have any Adnan's slides.
Yeah, when we look at very diverse, because it's easy to work with a small set of standards that you are familiar with their responses, and so we wanted to test the system, so we evaluated a large lean race collection from the Abiotic Institute, and so when we do that, we still see nice correlations of about 0.7 with the field, but you got to think, though, when you take any field environment and for rust and you want to correlate it with another one, or look at these data sets, you're probably going to have a correlation of about that anyway, because these resistances interact with the environment. So.
Okay. So we also developed some protocols to measure some agronomic traits in the speed breeding system. So this is actually Amy's work. What she did is she measured some traits in the speed breeding system, including days to enthesis, spike number, spike length, tiller number, and flag leaf length. And she tested the heritability of these traits using a panel, a double haploid population of 130 lines. And she tested at 2 different densities, a high density and a low density. So the high density corresponded to 12 plants in a 4-liter pot, and the low density to 2 plants in a 1.4 liter pot. And she actually found some nice heritabilities, especially for the high density, for spike length and days to enthesis. And then she calculated the genetic correlation with the yield. So she had 3 different environments, one with a low deficit, one with a post-enthesis deficit, and one with a pre-enthesis deficit. And she calculated the genetic correlation with the yield for different traits. They are listed on the right: days to enthesis, spike number, length, and tiller number. So this graph showed that there is actually some variation between there is some variation of the genetic correlation between the traits, but also for the same trait within environment. And this graph showed that tiller number is the one that seems to be consistent across the 3 environments, and that was quite well correlated with the yield, and that it could be used as the good proxy trait for yield. All right, now it's time to integrate all these phenotyping methods in just one generation. So this is Samir's work. He had a poster at the conference. So what he did is he used clear plots. He measured root traits. And then he spread the plants for relief rust, and then for crown rot. And then he measured the plant height. I don't know, do you want to comment about this? Or? No, that's all right? Okay. So I have this slide. Lee actually already presented it last time, so I'm going to go very quickly through it. But the idea was to show that using the speed breeding system or control environment conditions, if we can target multiple traits at the same time, we can actually decrease the number of plants. This is an example of breeding. One of the breeding company is using this system from 40,000 lines to only 100, and just a way to decrease the cost in the field. Yeah, so just quickly to finish, just wanted to present the work that we are doing with the accessions. We have about 300 land races from the Vavilov Institute. The lines were collected in 28 countries represented on the map from 1920 to 1940. The land races display a lot of phenotypic variation. We can see it from the spikes, but also we had an insight in the genes. This map represents, so we have only the 4 first chromosomes, but in black, it's the markers that are polymorphic in both the land races and a panel of Australian cultivar. And in red are the markers that are polymorphic only in the land races. So it shows that in theory we should have a really good source for new genes. And this panel has been evaluated for root traits. This is Annika work. The green dot, so this is the angle and the number. The green dots represent the land races, and the other dots represent the, some cultivars. But it just showed that the land races had actually a wider variation for both angle and number. The land races has also been evaluated for leaf rust in the field and in control environment conditions. And in both cases, we identified a certain number of accessions that had a high level of resistance. And this accession has also been evaluated for yellow spot, and same result. We had like a lot of land races showing a high level of adult plant resistance. I would like to thank my supervisors, but also all the PhD students that helped me to combine all the slides for this presentation today. Yeah, as I said, my PhD was funded by the GRDC, and if you want some more information on what we're doing with the speed breeding system, you can have a look at the Hiki Lab website. Thank you very much for your attention.